had a wonderful first 3 days of our workshop. First day we had introduction to systems biology and then whole exome sequencing, wonderful demo by Vandana. That was followed by Zone, who gave demo on RNA-seq, both using Use Galaxy. And even yesterday we had to use Use Galaxy from a wonderful talk by Akshay on single cell technologies, where there was single cell RNA-seq that was demonstrated. So today we have Akshay again, who will be speaking on metagenomics. So this is basically again a hands-on program. So Akshay will be joined by his colleague and employee Srinka Dutta, who will be giving hands-on and also a talk on metagenomics. There are students from Andhra University Vishakhapatnam here who are sitting in Sai Teja's account, and the others are the other participants. But I also recognize the presence of Rajiv Bandyopadhyay sir, my mentor, who is a professor at Burdwan University in Department of Microbiology. Thank you so much, sir, for joining. And of course, we recognize presence of few other.
Thank you, Prashant.
Yeah, thank you, sir. We recognize the presence of other life members as well. I could see Kalyani here and Sagar Kulape as well. Yeah, thank you. Thank you so much, Akshay, for your time. Please, please go ahead.
Yeah, thank you so much, sir. So today we will be discussing metagenomics and specifically microbiome data analysis. So initially I'll be covering the overview of metagenomics, and then later on my colleague Srinka will take over and she will do the demonstration on Galaxy. So here we will be using a particular data set. So that will be from cervical cancer, and we will demonstrate on Galaxy how to run the analysis and generate the insights for metagenomics data sets. Okay. So yeah. Okay. So I'm from GeneSpectrum Life Sciences, and this particular topic deals with microbiome to metagenomics. So we will delve over into what is microbiome, and then what is metagenomics, and when to use metagenomics, why we should use metagenomics, and obviously we will understand what is metagenomics in that. Okay. So this is what we are going to discuss today. So some introduction, techniques, and demonstration on Galaxy. Yeah. So this is a famous quote from Louis Pasteur, who is a world famous researcher, and he said that microbes are those who will have the last word. That means microbes were there, microbes are there, and microbes will be there. So everything that has happened from earliest days to right now, and or the present generation, and whatever is going to happen in future, microbes will be there to see everything or witness everything that is going to happen in this particular world, right? So that also suggests that microbes are very important in various different functionality, and that's why it's not like microbes got extinct, or they got mutated, or they got transformed into some other things. That has been consistent throughout the evolution. This is the tree of life that we have been seeing since early days as well. So here what you see is three different categories or three different branches of tree. One is for bacteria, other one is for archaea, and the third one is for eukarya. Right, but then in 2016, this interesting paper has been published in Nature Microbiology, and this is interesting because here what they have done is they generated this particular tree by considering all the microbes or all the bacteria which are being sequenced, right? So here you see that these are the bacteria, those are sequenced, and then here this is the particular section. These are the bacteria which are yet to be sequenced. So that is from 2016's perspective, 10 years ago. But then still what we can see here is the large number of section or large number of bacterial chunk has not been sequenced 10 years back. Few of them have been sequenced now after 10 years, but then still it is very large chunk, and that actually changes the view of life, which also considers that, or which infers that bacteria is something which is the largest branch of the modern tree of life, and this has made possible because of the sequencing, right? So going forward, as and when more and more sequences gets added, or more and more species get sequenced, we will have an elaborate structure where we might see bacteria dominating the rest of the categories. Okay. So what are microbes? We all know that microbes are tiny organisms. They have haploid genome, single circular chromosome is there. Usually there is no nucleus, and that's why we call them prokaryotes. And there is this easy interspecies gene transfer. Okay? So you can consider bacteria, archaea, fungi, protists, and other microscopic animals and viruses as microbes. Okay. So microbes are everywhere. So as we saw in the quote by Joyce Pascal, so microbes are everywhere, and all the animal evolution and development has occurred in the presence of microbes. But then if there are no microbes, then there will be horrible maladies for most animals. So microbes are very essential for development and metabolism of several organisms. And if these microbes are not there, then these kind of processes will be non-functional, and that will cause problems in various different animals. So in human itself, the gut microbiome that we have is a major driving force for most of the metabolic processes. And if we weigh the entire microbiome present in human gut, it will be it will weigh around like half a kg, right? And there are 3 times more cells which are present in gut microbiome compared to the entire human cells, or all the human cells which are present in a single individual, right? So it's very much important, and there are other consequences as well. As you can see here, the oxygen will not be there, and most of the ocean microbes alone account for half of your oxygen, and that's why it is very important to understand how microbial world behaves and what are the different communities which are present in microbiome. Okay. So now, as I said, the term microbiome. So let's understand what is microbiome. So this is basically an environment where you will find genomes of different organisms, different microorganisms. Okay. The main criteria to consider a particular microbial environment as microbiome, that it should be a mixed community, right? So what is a mixed community? So that means there should be multiple different types of microbes present in that particular environment. Then only we will call it as a microbiome. So, if there is bacteria A, B, C, D, E, F, G, then we will call it as a microbiome. But then multiple cells of a same bacteria, like if bacteria A is only there in all the environment, then we will not call it as a microbiome because that's not a mixed community. Okay. So microbiome definition is very simple. It's a microbial environment where you will find a mixed community of different microbes. Okay. So now, why we need to study microbiomes? So here, so there are several applications of microbes, right? So from environmental science to industrial applications to human health, everywhere you will find microbes are being utilized in one or the other ways, right? So if we understand in a specific environment, so let's say if there is some wastewater treatment we need to do, or we need to work on some wastewater treatment related things. So if we get to know what are the microbes or what is the microbiome composition of that particular site, then it will help us understand more about the site, and that can be further elaborated to have specific measures taken to perform wastewater treatment or any other different process, right? So, in context of human health, this is very important because uh, we get protection from pathogens, and also there are different microbes which are present in gut, which helps in absorption and production of nutrients. So that's why it is very important to understand the composition of different microbes present in different environments. So if we look at the human body itself, these are the different places where microbes can be found, right? So from like cheek to nose to like gut and intestine, and any particular body, like any particular body part, even on skin also we will have different types of microbes. Okay, so in general there are these four most abundant phyla which we see in human microbiome, and if there is any imbalance in these microbes, then that will cause dysbiosis, and it will lead to different conditions and different other disorders maybe. Okay. So human microbiome is very interesting thing, right? Because it can kill us with kill a specific person by acute infection, or it can also prevent the same infection. So this is a very interesting study that was published in 2011. So here there is one specific infection of C. difficile that causes severe diarrhea, abdominal pain, nausea, fever, and this is a recurrent kind of infection because even though the treatment is done with certain set of antibiotics, the spores remain in the gut, and later on these spores germinate to cause the infection again, right? So, as this is a recurrent infection, many times antibiotics will also have limitations, and there is a resistance that happens. So, what researchers found out is that, or what they performed here is that they performed a method called as FMT, fetal microbiome transplant. So this is a technique where a fecal sample from healthy donor is taken, and that is administered to the person who is infected with this C. difficile through enema or any other methodologies. And then later on, what they understood or what they noticed is that the infection has gone, and the person has got a healthy microbiota, which is actually fighting the infection against this particular disease or this particular bacteria, right? So this is a case where we can affirm that microbes are very important, and uh, if we understand their community, or if we understand their composition, then we will have better understanding of the further aspects like therapeutic aspects, or how we can improve the microbiome if there is any dysbiosis, how we can balance the microbiome and so on using prebiotic, probiotics and etc. Right. So as I said, dysbiosis is something which causes uh, imbalance, or if there is imbalance that causes dysbiosis, and through gut microbiome or through microbiome studies we can understand where is this dysbiosis happening. Right? Okay. Now, there have been different techniques to study microbial communities or microbiome. So, there are studies like rapid culture methods or culture-based techniques are there. Cultivation independent methods are there. Also, there are MS-based methods are there. Molecular techniques are there, PCR, real-time PCR, etc. And now we also have NGS-based methods to detect or to identify these different microbes. Okay. So, so when we talk about these different methods, there are some limitations of each and every method. If we talk about this culture-based methods, the major limitation is that only 1% of the bacteria which we know are, or only 1% of the bacteria, total bacteria, can be culturable. And these are the bacteria which are known to cause human infections or infections in other animals. And that's why people made efforts to make them culturable and prepare their cultures, right? Other remaining 99% bacteria, as they uh, uh, they have not shown any kind of pathogenicity to human or any animal. So that's why researchers or people haven't tried culturing them. So to culturing, we can only identify just this 1% of the bacterial communities which are there, and we will miss out this 99% of the communities. That's where metagenomics comes into the picture. So what is metagenomics? It's the study which involves analyzing genetic material obtained directly from environmental samples such as soil, water, human gut, or any other kind of sample without the need to isolate and culture individual organisms. So that is the key here. We don't have to culture any particular organism. We directly use the genetic material to identify what different types of microbes are present in that particular environmental sample. Okay, so this is actually done with the help of DNA sequencing methods, and we'll see how and what are the methods that can be utilized to perform this kind of experiments. Okay, so now we know that why metagenomics is important, so what is metagenomics and why it is important. Let's understand what are the common questions that are there which we need to address. Okay, so whenever we are performing metagenomic studies, these are the questions that so these are the questions for which we will get the answer. So the questions are like what microbes are there, what is the genetic diversity of this microbial community, then what functions are performed by this community, and then how do these microbes interact, and then what is the impact of this microbial community on human health, agricultural productivity or environmental sustainability. Okay, so these are the different things, or these are the different questions we can address by understanding the microbiome of any specific sample, right? So now we know that we need to answer these questions, but then how are we going to answer these questions? So we can analyze the genetic material, and then through different bioinformatics analysis steps, we can address all these questions, right? So that's where metagenomic sequencing comes into the picture, where we can sequence the entire genetic material present in the microbial community, and based on the specific type of genetic material we are trying to we are sequencing, we can understand more about that particular community. Okay. So there are two key methods in metagenomics, or two key approaches in metagenomics. One is the targeted approach, and other one is the shotgun sequencing approach. So both of these approaches are important uh, to understand microbiome or microbial communities. The difference comes into the region that is being sequenced. Okay? So in case of shotgun sequencing, uh, we obtain the complete genome sequence of all the microbes which are present in a particular sample, and then we go for sequencing, right? And then we further understand their composition. As opposed to that, in targeted sequencing, we only look for a specific region of interest. Okay, so for example, it could be 16S rRNA or 18S rRNA for fungi, etc. Okay, now depending on their usability or their use cases, people make use of these methodologies, and also both of them have their own pros and cons. Okay. So targeted sequencing is it's normally is cheaper because you are sequencing only a smaller region of a genome. It also provides great depth. It is precise, and it can be used in a routine practice. Okay. On the other hand, shotgun sequencing, here we are sequencing the entire genome, so that means it provides greater resolution at a very longer stretch of a DNA, and here you can also identify uh, the functions. Right? We can assess the function of the microbial community. Here we can identify novel organisms, genes, genomic features, etc. Right? So targeted sequencing, normally if we are doing 16S rRNA, then we can only find out the bacteria or the bacterial communities present in that particular sample. But here in shotgun sequencing, as we sequence all of the microbes which are present in that particular sample, we can identify bacteria or fungi or viruses or even other microbes as well. Okay, so that is the benefit of shotgun sequencing. Now, when we say that there are several benefits, but then uh, shotgun sequencing comes with a higher cost, right? As we are sequencing the entire genome of all the microbes, the cost is obviously very high. Also, the data is complex, right? So the data analysis infrastructure, or maybe the computational requirements are also very high for shotgun sequencing. Compared to that, targeted sequencing is cheaper and it can be easily analyzed. But then the drawback is like we can only look at one part of a gene, and the resolution is not enough. Okay, so we cannot identify species and subspecies from targeted sequencing. At the most, we can get 100% resolution up to the genus level. Sometimes, in some cases, we may get species, but then subspecies or substrain level identification is not possible with targeted sequencing. Okay, so these are the two approaches. Overall, considering the cost and usability and the applicability in modern day science, targeted sequencing is preferred. So you can also get several commercial applications of targeted amplicon sequencing uh, uh, of 16S RNA specifically. So there are several diagnostics kits which utilize this 16S rRNA targeted sequencing to understand the microbiome composition, let's say from human gut or maybe from the oral microbiome or any other site. And based on that, people are now recommending different diets and other different foods which can improve the microbiome, etc. Right? So here in this session, we will be discussing on the 16S rRNA sequencing data analysis and uh, the the sequencing protocol itself, right? Okay, so 16S rRNA is basically a component of ribosome. So ribosome, we all know, these are complex macromolecules, and these are also site for protein synthesis in all the living cells. Generally, there are two subunits of ribosome. In prokaryotes, uh, we have uh, 70S subunit with 50S and 30S subunits, right? In the 30S subunit, we have this particular gene, that is 16S rRNA gene. Okay, so now this why this gene is very much important, and why this particular gene is only used for metagenomic studies. Okay. So the reason that this gene is important, because it has something called as hypervariable regions. Okay, so hypervariable region is actually uh, uh, is a non-conserved portion. So this particular gene has both conserved and non-conserved sequences, but then these non-conserved portion has different types of polymorphisms for different taxa, right? And that is what is exploited or utilized to understand or to identify different microbial or bacterial communities or bacterial classes based on this 16S rRNA gene. Okay, so there are 9 such hypervariable regions which are there, V1 to V9. Okay. So the typical length is 30 to 100 base pair, and each of these regions have specific function, or each of these regions have a specific resolution to different taxonomic levels. So, for example, the V4 region is semi-conserved, and it is mostly used for resolution up to the phylum level. Okay, so if someone wants to identify bacteria up to the phylum level, then we can use this thing, V4 region, for sequencing. V1 to V8 regions are of pathogens, and then there is this V3 region which gives you genus level differentiation, right? So V4 is giving you phylum level, and V3 is giving you a genus level. So normally what people do is, like, they sequence both V3 and V4. So that reduces the cost, as well as it gives you up to the genus level differentiation of bacteria. Sometimes people also sequence the entire V1 to V9 region to get more detailed insights about the bacterial communities which are present. Okay. So for each of these regions, there are universal primers which are available, and these anneal to the conserved regions, and based on those primers, the sequencing can be performed. Okay, so now let's see how the data is analyzed. I'll just give you an overview, and after this, Srinka will take over to the Galaxy-based pipeline for analyzing 16S data. Okay, so normally 16S rRNA sequencing is actually a DNA sequencing, right? So we are sequencing the 16S rRNA gene, that is a DNA sequence. Normally it happens that in DNA sequencing we look for genotypes, but here we are not interested in genotypes, but we are interested in taxa. Okay? So as we want to identify different bacterial compositions or different bacterial communities, we need to look into the taxa. So there are different ways by which the taxa can be grouped. You can have taxonomic classification, phylogenetic tree, or someone can use OTUs. Right? So OTU is basically operational taxonomic unit, which presents or which represents group of related organisms. Right? So so let's say if for a specific species there are different OTUs, right? So that represents the specific species or that particular genus. Let's say a genus has different OTUs which are representing different species. Okay. So so how this is done in metagenomics? So what we do is like we identify these OTUs from the raw sequencing data, and we map the OTU sequence against a reference database sequence. So a reference has already signatures for different bacterial classes. Okay, so we map those signatures with the OTUs that we have obtained, and if the signature matches, then we can say that this OTU belongs to that particular class of a bacteria. Okay. So this is how the analysis is performed. So we get the raw data from sequencer. We need to sort the reads. Then there is some quality control that we need to perform. And then after that we go for trimming and filtering. Okay. So here we remove those reads where the quality is low, or where the bases are above or below the given quality. So we trim and remove those kind of bases from both the reads. Then we need to go for dereplication, error correction, and then sample inference. So after sample inference, we get a OTU table. So OTU is nothing but so here in Data 2 we call that as ASVs, right? So ASVs and OTUs are same. So we get this sequence table. Then there are some processing steps that we need to follow, like chimera detection, and then we go for taxonomic classification. So here is the step where the OTUs are mapped to the existing sequences in database, and we infer the taxonomy. Okay, and then later on we go for diversity analysis and visualization. Right. So all of these steps will be discussed in detail in the next session taken by Srinka, and we will then discuss or we will explain what is chimera detection and what is diversity analysis and so on. Okay. So that's it from my side. If there are any questions, we can discuss those questions. Otherwise, I'll hand over to Srinka. Are there any questions?
Hello, sir. Am I audible?
Yeah.
Okay. Sir, my question is, does each individual has different microbiomes?
Sorry. Can you repeat your question?
Like, Each individual has different microbiomes. Like at the individual
Yeah.
level, does this
So
microbiome
microbiome,
varies?
yes, absolutely. The microbiome composition will be different for every organism. See, ultimately it depends on what you eat and what is the environment that you live in. So based on that, the composition might change, right?
Yes. So then you have mentioned a technique called FMT, that is fetal microbiome transfer.
Hmm.
So how can we transfer the microbiome of one individual to the other
Hmm.
when each has
So
different
yeah.
microbiome composition?
So basically, the FMT is a method where microbiome of a healthy individual is taken and that is transferred to a person who has infection, which is a recurrent infection, not getting cured by antibiotics and other treatments, right? So
Mm.
it's from the healthy individual. So obviously the composition will have all the good bacteria, right? So it's
Yeah.
not like that each individual has different microbiome composition, but
Mm.
those microbiome or those bacteria will not be of use, or those will be unhealthy bacteria itself. There can be healthy bacteria as well in composition, and
Yes.
then the difference might be there, right? Yeah.
So I have one more question.
Hmm.
So you have discussed about the sequencing in metagenomics. We sequence uh, we sequence the whole microbiome. So what if
Hmm.
we found some sequence which of the organism which has not been found yet or not has been discovered yet? So it may be quite possible that in that sequence we have some sequence that we have not found that sequence in any other microorganism.
Sorry, I couldn't get your question. So you are saying that if a sequence is not found in other microbes,
In
sorry, I
metagenomics,
couldn't.
we sequence the whole of the genome of a community of the whole microbiome, right?
Hmm.
So can we get a
Yeah,
sequence which has not been discovered yet, like the
yeah, that
bacteria?
is possible.
Yeah.
Yeah, that is possible. So if you are doing short-term sequencing, then you can identify those novel organisms and sequences, right?
Hmm.
With 16S sequencing, it is sometimes difficult because it relies on the existing information, right?
Yes, sir.
As opposed to this short-term sequencing, where
Hmm.
you are trying to identify novel organisms, or
Hmm.
you are trying to find out the microbes based on the entire genomic composition, right? So
Yes, sir.
there you have this great chance of finding out novel organisms, or which are not discovered earlier.
Okay, okay. Sir, I have one more doubt. So you have discussed that
Kushbu,
we have
excuse me,
16S
Kushbu.
RRN.
Excuse me, Kushbu. Can you please switch on your camera and kindly ask question,
Yeah, yeah,
please? Huh? That
yeah,
will,
sure, sir.
that's a minimum etiquette, in my humble opinion. I'm really sorry for this. Okay, please.
So no problem. Is it okay now?
Yeah,
Okay.
and please introduce
So,
yourselves, huh? Please, huh? Thank you so much.
thank you, sir. So you have discussed about the 16S rRNA, and you have told that it has hypervariable region that is non-conserved portion. So, sir, I have a doubt that if it is non-conserved portion, then like if it is not non-conserved, then it can vary from organism to organism, right?
Yeah.
So, how we are using this hypervariable region to identify the bacteria when it is not conserved?
Yeah. So basically, hypervariable region is uh, so when we uh, uh, use that particular region. Okay, let's frame this in such a way that you have this different region, which is
Hmm.
which holds signature for a specific type of bacteria, right?
Yes, sir.
So as I said, we have like V3, V4 regions. So V3 gives us resolution up to genus level. So
Yes,
there are these signatures which are specific for a specific bacteria,
specific community.
and
Yes, sir.
yeah, and these are there in the 16S gene, right?
Okay.
And we utilize those signatures here when we sequence the specific region of the 16S rRNA gene,
Yes,
right?
sir. Like,
So
I just
based
want
on
to know,
that,
like,
yeah.
we have the reason, different reason, like VT before and so on. So I want to just understand if it is non-conserved portion, then it may changes, uh, like after one or two generations or maybe after some evolution. So how, if it is non-conserved, then how
No,
we are
no. So that's
using?
yeah. That's the thing. Uh, it is a region which has not changed specifically for a specific bacteria throughout the evolution,
Okay,
right? So
okay,
there are polymorphisms which are present among various like among the organisms of various taxa in these hypervariable regions, right? But then those polymorphisms have remained same throughout the evolution. So there is very interesting article which is there on this particular 16S rRNA gene, and the first researcher who identified this particular gene uh, actually showed that throughout the evolution there are these specific regions or these specific polymorphisms which are not changed, right? So that is what is being exploited here.
utilize. Okay, okay. Thank you, sir. Thank you so much.
Maybe Akshay, you may want to go
Can I
with Kalyani first, Kalyani or Sharanya maybe. Yeah, please.
Yeah, so there are Sharanya has this question: what is the difference between ASV and
So
OTU?
Sharanya, you can switch on your camera and ask if you want.
Yeah, so ASVs and OTUs are almost same. When we use data pipeline, it generates the ASVs, right? And in general, OTUs are the operational taxonomic unit of any particular phylogenetic tree, right? But then OTU sequences, sometimes some other tools may give you OTU sequence. Specifically, we are here using Dutta, which gives us ASV sequences. You will get to know this more when Srinka will explain this particular part. Yeah, again, OTU will explain again. Kalyani has this question. What all sample types are generally tested? What is the concentration threshold for various bacteria found, and is there any specific standard followed? So you can sequence all the different types of samples. It can be soil sample, environment sample, human microbiome, animal microbiome, plant microbiome, any kind of sample wherever there are microbes present. Uh, Concentration threshold is as such not there, but then what we have is the identification levels. So depending on the region that you have used, you can find out the specific, like you can have the specific this thing uh, uh, classification of the bacteria identified at a specific level, right? And any specific standard followed. So standard in the sense uh, there are some known studies which are found to be or there are some known studies which says that a specific type of bacteria is found in a particular type of microbiome. So if that is there, then we can compare that with the existing data and the experimental data that you have, right? Uh, Sequencing standards are always followed, and many times, like selection of the region is something which is also depending on the site which we are working on. Okay, next is sometimes people go for targeting various regions like V3, V4, or in some cases V2, V4. What is the difference in there? Yeah, so I told you, right? So each of these variable regions have different levels of identification, bacterial class identification. So V3 can identify up to genus level, V4 can identify up to phylum level. That's the reason that people use V3, V4 many a times, or in some cases V2 to V4. Sometimes the entire region like V1 to V9. Again, it depends on the cost, because sequencing a larger DNA is costlier compared to a smaller DNA. So if many times the purpose is to identify at a genus level, so V3, V4 is sufficient for that. Yes. How much error rates are valid or acceptable when using data pipeline? So actually, in data, it corrects the error, right? So we don't have to select the threshold, and based on that, we don't have to accept the error rate, but actually it corrects the error looking at the existing data which is there. Okay? So it runs a machine learning based model to correct this kind of errors in data pipeline. Yeah, so each region have different polymerase caves. See, we have several databases to identify bacterial classes, like there is Silva, RDP, or Green Genes. These are the three commonly used databases. Now, if there is anything which is not covered in the database, we cannot identify using 6-mess rRNA sequencing approach. Right? But then these databases covers most of the well-studied, commonly studied and unstudied organisms or bacteria. So there are very less chances that you will not find any specific bacteria when using these databases. If it is like extremely uh, uh, extremely different samples, uh, so there are chances that there will be novel bacteria in that particular sample. So in such cases, you should go for shotgun sequence. How can we define a novel bacteria? So anything which is not identified earlier can be called as novel. There are some instances of microbiome analysis where some of these novel bacteria can be identified, right? So, or even if it's not the entire new species of a bacteria, but you may find out some subspecies or some strain which is not previously identified. Sometimes it may also be possible that the strain is there, but it was not sequenced earlier, that you can identify and that you can sequence and identify the this thing. Yeah, so
Yeah, so is there any way to, like, when we are defining any novel culture or novel bacteria, so how we can say this is novel based on only the sequence matching, or is there any identification body in NCBI or any other way where we are, like, let's say
yeah.
I have a sample and I have given this is a novel bacteria or novel species. I don't know the phylum is related, where is the species. Then the nomenclature, how we define the nomenclature, how we submit that sequence to the NCBI and just like define that is novel. So there should be some process. I'm not aware about that, but maybe if you are, then it will be good for.
Yeah, so normally when we go for data analysis, uh, so for shotgun sequencing, we normally perform like the variant calling process or variant calling workflow, and then we also need to perform the assembly, right? The genome assembly. Post that, we need to perform the annotations, genome annotations. So if there are any specific sequences which are getting annotated or which are not getting annotated, that we can flag and identify from this whole data set that we have. Now, based on the sequence similarities, we can identify the genes. If some of these genes are not matching to the process in which we expect that the bacteria should be involved. See, if you are taking gut microbiome, so I'll take an example of let's say a gut microbiome study for um, uh, for a for a for a pregnant lady who is delivering a preterm baby, right? So, uh, there are normal babies, full term babies, and then there are preterm babies. So, what are the differences in the microbiome present in both of these uh, both of these like individuals, right? So, if we find some kind of uh, bacteria or some kind of organism that is not commonly found in the normal scenario, right? So that can be a novel for a scenario where it is giving preterm birth, right? So this can be gone through various different annotation pipelines and uh, uh, your sequence matching pipelines, and based on like microbiologist understanding, this can be flagged as a novel organism. I am not a microbiologist by training or by experience.
Okay, I think I got your point. So basically,
Yeah.
if we are getting any noble strain in our analysis, definitely we have to recheck it. And just when we are having that strain, I think we need to go for the assembly and all those things and define the everything to that. Yeah, my question was specific, like, is there any body who is checking these novel strains, which the people are submitting to the NCBI? Definitely
Sorry,
NCBI
I
will
am
have
not.
that regulatory. Yeah, that's okay. That's okay. I think I got the point there. Yeah. And another question I just want to understand whether we are having any multi-agentic system based analysis for the metagenomic sequences yet.
Uh, I don't find any of, or I don't remember any of the systems which are there. Uh, Although there are some pipelines which can work as a plug and play kind of things, but
Okay,
agentic
got it. Yeah,
pipelines
got
I
it.
don't know.
Yeah, I think it's a very evolving area, and people may are working on those kind of the development. Yeah, definitely. Yeah, we'll let. Yeah. Nice.
Yeah.
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 Yeah, I know you must be tired, Akshay. Thank you so much, for answering so patiently all those questions.
Yeah, no problem at all.
I
I
just
think
have one question,
questions are
Akshay.
very.
I just have one
Yeah.
question. I'm very curious. The last 1 or 2 years, I've been, pervasively observing many authors claim that if there is a tissue metagenomics study that they wanted to study, they would only take or analyze metagenomics from that particular tissue, but they claim that the gut microbial metagenomics is also adherent to those kind of tissues, which means if I would like to analyze, say a prostate, what do you call, gut microbiome metagenomic analysis, they say that the normal gut microbiome microbial data can be translated to the knowledge that is already there with the prostate gut microbiome data,
Yeah.
which to me is bit, I mean, I was also scratching my head, why, how is this justified? I really don't understand. I mean, is there a way? I mean, can we say that? I don't know.
Oh yeah, I mean there are several studies, and in practice also many times we see that for let's say for this thing, if there are any infections in women, like urinary tract infections and so on, normally people recommend prebiotics and probiotics to improve the gut microbiome as such. So overall, it's all like gut-brain axis which functions, and that's how it's related. I was interviewing one of the candidates today, and she was working on the role of gut microbiome on Alzheimer's, right? So she found some kind of proteins which are being secreted by gut microbiome, and those are associated with the progression of Alzheimer's. So that can be absolutely possible.
Yeah. So we have better bioinformatics algorithms also to detect such things?
Yeah,
Okay.
we now have very like uh, matured pipelines which can
Okay.
detect such kind of.
Maybe I'm not agreeing with this fact. We can correlate the like how the gut is behaving uh, to maybe to decrease or increase of certain disease, but we don't have till now, as per my knowledge, we don't have any certain proof. Some of the proof is there, but certain proof for Alzheimer or something like that, I as per now, as per day today, I'm not aware of that. If it is such fact or proof are there, then I think we can say like, yeah, this kind of the microbiome are treating for the cancer, treating for the Alzheimer or something like that. Am I?
It's not a treatment. I guess you are
Yeah, it's
misunderstanding this.
maybe
It's the
increasing
role
the.
of microbe
Mm.
for a particular condition, or how the microbes change overall biology and physiology of a particular condition. That is what we are discussing here.
Yeah, I got it. That's why I'm telling like the how the microbe are changing the physiology or that's again uh, if metabolism process are good in the human body, maybe it will helping to the curing of certain cancers or something like that. Correct?
I still don't have any kind of research in.
Yeah, that's why I'm telling you such research
So
is
that's what
evolving.
I am also telling. It's not curing anything. It's the association of gut microbiome with other biological functions of human body.
Yeah. Yeah. Thank you, sir. So I was just wondering, since you also work for a company, does your company do this sort of?
Uh, no, basically we are totally in the software development and agentic based system development to our clients, and people are, yeah, so we used to develop ML models, even the bioinformatics pipeline and all those kind of the things we provide to our clients. So those kind of, so we are not basically in the genomics and doing some sequence analysis, something part that, but definitely our more main motive is like development, most of the agentic system and all those. Currently, my team is working on that. Yeah.
Right. Yeah. Thank you. Yeah.
Yeah, great. All right. So I guess I'll hand over to Srinka for the next hands-on thing. Yeah.
Thank you so much, Akshay, for the excellent foundation. So I hope everyone has now the knowledge about what metagenomics is and how sequencing in metagenomics is performed. So now, for the next 1 hour and so, I'm just going to take on what Akshay has explained and show you the demonstration about how actually we do the analysis and what is the conclusion we get from the data analysis. So let me quickly share the screen. Okay. So, before resuming with our demonstration, I'm just going to quickly show you uh, the specific study we are going to analyze today and the biology behind it. Right. So, the study we are working on is about cervical cancer patients and healthy microbiome of the healthy womb. So, why this study? So, why this study? Because we healthy microbiome, vaginal microbiome of healthy women is seen to be dominated by lactobacillus. And predominantly by lactobacillus, and this particular genera produces lactic acid, which is known to maintain a low pH and inhibit the pathogens. Whereas in the patients with cervical cancer, it is found that this particular lactobacillus bacteria, lacto,
Srinka, may I know if you're sharing your screen?
yes, I'm sharing. Isn't it visible?
No, it's not visible.
Just a second.
It's visible here, Kalyani.
Yeah, I can also see that.
Here we can see that. Yeah.
Okay.
Maybe
Not
that is
able to
because
see
you have to go up, Kalyani. Maybe I was spinning, Now, how about now? I think you can see that. Yeah.
Yes, I'll just unpin her. And now, can you see that? Can you see the screen?
No, the screen is not visible.
So maybe you can rejoin the meeting because it's visible for most of us.
Sure, sure. Yeah. Sorry for the interruption.
Yeah, so basically, in this demonstration, we are just going to see how the bacteria in a healthy woman is different from the one in cervical cancer patients, which is known to have a dysbiotic microbiome with other anaerobic species in rise, and the lactobacillus genera is very much depleted in cervical cancer patients. This is also been reported by Leon Gomez and Romero in their study, where they studied 12 papers on this correlation, and they have also been they also suggested cited that they have seen this kind of dysbiosis in most of the patients. So this is the study we are going to see perform. So this was a study that is been published by Fan and his colleagues in 2021 in a journal called Cell Death and Disease. It enrolled 119 women, from where this 54 are healthy controls and 65 are cervical cancer patients. So they have collected their vaginal swab samples, and they have sequenced the V3, V4 region, just as Akshay covered. This region is commonly used region for bacterial identification in microbial research. So, why this is interesting? Because, as I have said, the healthy vaginal microbiome is actually unusual in microbial world. So, if you consider other samples like soil samples we have, then bone samples from any water bodies or gut microbiota. There you can find that it is a mixture of microbiomes. But in this case, the vaginal microbiome is predominantly they have lactobacillus genera, right? So in this study, they have seen that this lactobacillus, particularly lactobacillus inners have been dramatically depleted. And what does this lactobacillus inners do? It produces lactic acid, which in turn suppresses the WAT-FUT8 signaling pathway. And this pathway is known for its tumor generation, right? And when the lactobacillus inners bacteria is depleted, it is correlated with tumor formation and ultimately the cervical cancer. So that is the main motive of this study. So basically, the study have 119 samples, from where for the demonstration purposes, I have just taken 10 samples, 5 from healthy controls and 5 from cervical cancer patient, as we are going to demonstrate in Galaxy. So this samples, I have subset the samples accordingly. And this is the project ID, bioproject ID for the study. You also can search this in NCBI and read about the study more if you want. Also to be noted that these samples are single-thread FASTQ files, and mostly if you go on studying metagenomics, you will find paired data. But this is particularly a single and FASTQ. So there are a few steps that we can skip that I will eventually tell you. Also, for reference database, I have used Silva 138 database, which is the latest Silva database. And Silva is mostly updated database, so we'll use this. So this is the pipeline we are going to follow for that is the data to pipeline in Galaxy. So we are going to start with raw FASTQ files that I have shown here. I have the FASTQ files of these samples. We are going to start with the raw FASTQ files, and in case of like paired data, we need to sort out the front and reverse reads of the paired data in the Galaxy itself. So they have in Galaxy. They have this automatic system they use for handling the forward and reverse reads. So like basically we just need to upload it and form a collection of all the forward reads and reverse reads separately, and then we will be going through each and every step for those reads individually. Then here in the merge read section, we are going to merge it just before constructing the sequence table. We are going to merge the forward and reverse read of all the samples, right? After sorting, we have quality control. For that, we use this data to plot quality tool, plot quality profile. So after we run this tool, we get a graph of red score where we can see what the quality of each base position is, and we can then decide our trimming length, for mostly for paired end data. We would run this separately for forwarded reverse collection, as I have mentioned. But for our single end data, we are just going to run it together. So after that, after analyzing what is the quality, we decide to trim it. So mostly in metagenomics sample quality, it is seen to the samples have degraded the quality towards the end of the base. So as per my knowledge, I have noticed this pattern in most of the metagenomics, like all of the metagenomic studies I have done till date. So according to that, we will need to trim that up to that base pair where from where the bases starts to get depleted, right? So then we have dereplication, where that is handled in the background only, in the error correction step. So here what happens is that the similar kind of sequences are clubbed together so that the process becomes faster. So again, we have error correction where we create a model for the error by analyzing the sequencing error they can perform. So this is the most distinctive step in Data 2. So every sequencing run has its own characteristic error profile, right? So its own pattern of miscalls. So learned error, here for error correction, we use a tool called learn errors, right? So learn errors builds a statistical model of those errors from your actual data that you have. So instead of applying a generic quality filter, Data to learn the specific mistakes your sequencer made on this specific run and then corrects them. So this step is error correction is the step where we build a model. Then sample inference is the step where we correct the error, right? Using this model that we have created from error correction step. So after that, for a single end data, that is for our data, we'll skip this merge read step because it is used only for paired. Then after that, we are just going to construct the sequencing table, right? Then what we are going to do here in the sequence table, what will happen here? Just let me show. Here we'll get this table where we have samples here and we have this ISBs here, right? ISBs here. So we'll have the sequences here, and how many times this ISB have mapped the number here, right, for each samples. So we'll get this type of data here, table here. Okay. Then we have chimeric detection. So chimeras are some artificial hybrid sequences that forms when two unrelated DNA fragment joins during PCR amplification. This is kind of a like mistake while PCR amplification. So like you can call it as a lab artifacts. So if we don't remove it, they might inflate our diversity estimations and pollute our taxonomy results. So this step is also a very important step. Then next we go for taxonomic classification. Here, this is the step where we assign those ASVs with the actual bacteria name, right? Then we'll after this we are going to have the table where we have the samples, where we have the ASVs and the bacteria, like from starting from the kingdoms till genus for 16x. And nowadays, sometimes the species level is also mentioned, but it is very rare. Okay, so up to genus level, like we get the data. Then we have diversity analysis and visualization. Here we study alpha diversity, beta diversity, and the taxonomic like bar chart of the of this taxons, like how many like what is the amount of what is the amount of this lactobacillus present in a particular sample, that which is our main motive of the results. So that is what we are going to study here, right? So this is particular this is the workflow we are going to follow for our demonstration. Next, we have what we can expect to find, like the last step that I suggested, that is the conclusion we get from the study. So we are expected to get these results as from where we can understand these biases in the microbiome. So we have alpha diversity. So in alpha diversity, we see the population within the samples. Okay, so here you can see that like for, as I have said, that in healthy controls we have lactobacillus dominant microbiome. So in healthy controls for alpha diversity, we are expected to see low diversity, whereas for cancer samples, we are expected to see higher diversity of the microbiome. So in beta diversity, we mostly see how the microbiome is different from the like samples. Between the samples, that was this alpha diversity is within the samples. In each samples, how it is diverse, and beta diversity means how the samples are differs in according to the type of sample, right? Then we have the taxonomic plot that I have told you, that will just show which bacteria are predominant in the samples that we have, that is healthy and cancer samples. So pretty much this is what we expect. So next we have, why does it even matter, right? So from when I was going through the study, I have told you about what happens when this lactobacillus inert depleted, because this lactobacillus inert helps in maintaining a low pH, because it produces lactic acid, which in turn suppresses this signaling pathway that is associated with tumor formation, and ultimately the cancer cells are suppressed. But without lactobacillus, the pathway is not suppressed, and as a result, tumor glycosylation proceeds, and there is probability of cancer progression, right? So if like there is an early detection, then this cancer cells, the cancer progression can be prevented with multiple like clinical implication, like probiotic therapy. Even Akshay has also highlighted that. We have probiotic therapy. Then we have this from the study. We also have this biomarker potential, like this anaerobic species that are predominant in the cancer patients, like uh, Gardnerella and Prevotella, is also identified as key microbiome biomarkers for HPV infection and cancer progression. So there is this study also, like from Leon Gomez and Romero, that have associated HPV infection and cancer progression as well. So, like these days, HPV vaccination is also in very much demand, and even everybody is promoting the women to get this HPV vaccination, because it is also associated with the cancer progression. So these are the clinical implication that is associated with this microbiome and metagenomic study. So next, let's start with the demonstration. Before that, I'm just going to show you the samples I have. See, these are just FASTQ files we have, and also we need this metadata. That is the information about the samples. So I have made this metadata table manually itself, because I have a limited number of samples. So these are the samples, 10 samples. I have used a column called group to group it. It is like used while building the graphs, the bar plots in for taxonomic analysis. And then we have the SRR ID and the subject ID. These are optional. Like you can also use this to like in your x-axis or y-axis for your bar chart. So these are the this is the metadata that is used. So for this, I'm just going to
Akshay. Hello.
log in.
Hello.
Yes, Yashita, do you have any question?
Yes, ma'am. I was asking if you could please share this raw data files with us, or some links
Akshay,
through
I'm
which we
sorry.
can also
Akshay,
access
can
it.
you
Yeah.
record this demo?
Yes,
and send it
I
to
have
me?
mentioned
Can you
this
do that?
project ID in the PPT. Let me show.
You know,
So one with
yeah,
this,
record the screen. That's it. Yeah, please. Only this record.
yeah, this is the project. Let me share this in
Yes, please.
chat
And
as
then,
well.
yeah.
So when you search this project ID in NCBI,
Mm-hmm.
you will get the information about this.
Okay.